Tuberculosis is a major health threat, and everybody agrees that we need a good vaccine to control this devastating disease. You may ask, do we really need a vaccine? After all, we have BCG, Bacille Calmette Guerin. This vaccine indeed protects young children, babies, from tuberculosis. However, it fails to protect adults against the most prevalent form of this disease. So there are two avenues of new vaccine developments. Both trust on BCG. The first avenue are so-called subunit vaccines. This is a few antigens in adjuvant or presented by a virus vector, and they are given after BCG. The other approach is a genetic modification of the current vaccine BCG. This approach is followed by us. So we took BCG and introduced new genes into this vaccine because we knew that BCG gives some protection and we understand the immunologic mechanisms underlying. And by the genetic modification, we broadened the immune response so that we have a better armamentarium against tuberculosis. So our hope is that this vaccine is much better than BCG in protecting against TB. Moreover, this new vaccine candidate has proven to be safer than BCG. Now this has all been done in preclinical models. 3 years ago, our vaccine then also went into clinical trials. The first step is that you test this vaccine for safety and immunogenicity in adults in the country or region where the vaccine was developed, in this case Germany. That was successful. The vaccine proved to be safe and immunogenic. We then repeated a very similar clinical trial in South Africa in adults, again successful, safe and immunogenic. And then immediately we now went into newborn because that's the target population of our vaccine and this is how to do it. And again, this was done in South Africa and the direct observation is completed, and again our vaccine candidate proved in newborn also to be safe and immunogenic. The vaccine is now being prepared for a phase 2b clinical trial, and therefore here we will find out whether it also shows efficacy. This needs large populations because you look at vaccinated individuals, whether they are protected against the disease, and therefore compare these individuals with other individuals who just received the conventional vaccine BCG.